Thanks for remembering, Clarissa. So I am happy to introduce Luis Carvajal-Carmona, who he and I were just talking, met over 10 years ago through some of our joint collaborations with people in epidemiology. So he is now the Auburn Community Cancer Endowed Chair in Basic Science at UC California, Davis. He has multiple leadership roles there, but I'll just comment relevant to our forum that he's Associate Director for Inclusivity, Diversity, Equity, and Accessibility. And he's the founding director of the Latinos United for Cancer Health Advancement Initiative, as well as the Center for Advancing Cancer Health Equity at the UC Davis Comprehensive Cancer Center. And he co-directs their community engagement program, which NCI puts huge emphasis on the community engagement programs of their cancer centers. He's been awarded the UCD Chancellor's Award for Excellence in Mentoring Undergraduate Research, and also an Advanced Scholar Award for his outstanding scholarship. He's a member of AACR, AACR, American Association for Cancer Research. Those of you not in cancer, it's the largest cancer research organization in the world. Including the Minorities in Cancer Research Council, the Minority Scholars Award Committee as co-chair, as well as the Epidemiology Working Group. And he sits on the AACR Racial Inequities Task Force and the National Cancer Advisory Board Working Group on strategic approaches and opportunities on cancer, on research on cancer among racial and ethnic minorities. So he got his PhD. It's always good to introduce people. You learn things you don't know. From University College London, did postdoctoral training at the London Research Institute, and was a senior research fellow at the University of Oxford. And he's now PI and multi-PI of grants totaling over $17 million. So his research, as you'll hear, focuses, and that's why I invited him for our forum, specializes on cancer genetics, epidemiology, health disparities, and global health, and particularly related to genetic origins of the Latino population and unique genetic determinants, increasing the risk of colorectal cancer, and a novel familial form of gastric cancer. So with that, I will turn it over. And so Luis is going to talk about his genetics and how it relates to, his genetic research, but how it relates to using gastric cancer as an example of working in diverse populations for cancer that has a highly different incidence among different populations.
Okay, thank you, Sharon. Happy to be here. Thank you for the invitation. I am connecting from California. I've been here for 10 years, and other than some of the leadership positions that I do, I am a scientist, so a cancer geneticist. So I'm going to cover three main areas in this talk. As the title said, I'm going to flip the order of my presentation, and I'm going to introduce you to the epidemiology and disparities of gastric cancer, also stomach cancer. Some people call it stomach cancer, the same. Then I will present an overview of gastric cancer genetics, predisposition, and describe some of the work that we have been doing in the lab with regards to inherited forms of cancer. And finally, I'm going to be presenting data and results from studies on somatic genetics, on cancer genomics, and how intratumor heterogeneity affects, possibly affect responses to treatments. So let's start with epi and disparities. Gastric cancer is a really important cancer worldwide. There are 1 million new cases diagnosed every year in the planet, and around 730,000 people die because of gastric cancer. This is a very bad cancer to have. The prognosis is poor for multiple reasons. For one of them is most tumors are detected in late stages, and in later stages, the survival is 4 to 5%. The survival to metastatic gastric cancer remains very low. There are no well-accepted plans for early detection, right? Unlike, the 4 preventable cancers, breast, lung, cervical, and colorectal, there are no clear guidelines on how to prevent gastric cancer, even though 3 high incidence countries have plans, screening plans that offer endoscopy. starting at 14. Those are Japan, Korea, and certain parts of China, where the incidence is very high. In the US, is the leading cancer disparity in Latinos, and it's also very common in communities of color. And not only this cancer has a really bad prognosis, also there are very few options when the patients present with gastric cancer. There are only 2 FDA-approved targeted therapies, right? There is a third one that was approved recently, but that one is an improvement of Herceptin. And also there is some of them are eligible for immunotherapies. But in general, compared to other common cancers, there are very few molecularly guided therapeutic options. This is these are pie charts of incidence and mortality, of cancer mortality. And as you can see, the big 4 are the big 4: breast, lung, colon, and prostate. But the 5th most commonly diagnosed cancer in the world is stomach cancer. Okay. When it comes to mortality, it rises to the second to depending on the year, but it's around the 3rd, 4th most main cause of mortality. So as you can see, and we don't speak a lot about the gastric cancer in the US because the incidence is relatively low compared to other part of the country, of the world. But there are many regions in the world that are heavily affected by this. And if we were going to plot the number of cancers, gastric cancer in the world, China, Asia has most of them. So particularly China has half of the gastric cancer cases in the world, but also certain parts of Eastern Europe, Latin America, and some of other developing nations. So gastric cancer, as most cancer types, is a complex disease. Is more common in men, is more common in Latinos and population of color. It's a highest incidence in East Asia, South America, and Eastern Europe, as I showed before, and it has low incidence in North America and Africa. The main risk factor for gastric cancer is a bacteria that colonizes the stomach called Helicobacter pylori, and around half of the gastric cancer cases can be attributed to Helicobacter pylori. And this is a strategy that is being pursued, which is basically trying to eliminate Helicobacter pylori as a way to lower in gastric cancer incidence. As for most common cancers, smoking is a risk factor. A virus, Epstein-Barr virus, is also a risk factor for gastric cancer, and 1 in 10 gastric cancer cases are caused by this. It's a relatively late onset disease. The average age of diagnosis for gastric cancer is 70. And there are other risk factors in the diet. Reflux is also a risk factor for upper gastric cancer. And until recently, it was believed that only 1 to 3% of the gastric cancer cases were caused by inherited elements. That is being reevaluated with more recent data from our group and from other groups where we see a larger fraction of patients carrying germline mutations that can cause gastric cancer. If I were going to treat the epidemiology of gastric cancer, one of the most common way to kind of stratify is the location in the stomach. So this is a figure of the stomach, and the epidemiology differs on when the gastric cancer happens. If it happens in the upper part of the stomach, this region which is called the cardia, the risk factor profile is very different than if the gastric happens in the lower part of the body of the stomach. So for cardia tumors, upper stomach tumors, being white, is the relative risk for in whites is higher, around 4-4. Here is where the reflux, gastroesophageal reflux disease, GERD, is also a risk factor. So when the gastric acid flows up during reflux, that affects this epithelia and increases cancer risk, and it's also more common in men. So this is one of these cancers where it's more white men are a high risk. For the non-cardia, which is most of the rest of the stomach, the main risk factor is Helicobacter pylori, and the relative risk is between 6 and 21-fold. This is an important risk factor for gastric cancer. And also lower gastric cancer is more common in minorities, in part because they also have high incidence of gastric cancer, but also there are other social determinants of health that affect the risk. Oh, this is not a good slide. So this is a study published a few years ago where they basically look at SEER data, cancer registry data, and they found that basically in the upper gastric cancer is more common in whites, less common in minorities, and is not affected by socioeconomics. The lower gastric cancer types or non-cardiac cancer is rare in whites, is more common in minorities, but is heavily affected by socioeconomics. So if you are from a low socioeconomic group, your incidence is higher than if you are from a high socioeconomic group, which was an interesting kind of socioeconomic social determinant of health study on gastric cancer. So in addition to to being an important cause of cancer incidence and mortality worldwide, gastric cancer has a particularly high burden in minority populations. This is the data from in the US from SEER in all racial and ethnic groups, men and women, and this is incidence. Okay, so as you can see, the incidence of gastric cancer in whites is around half of the incidence of gastric cancer in Blacks, Asian Pacific Islanders, and Hispanics. Okay, so in general, minorities are between 1.5 to 2 times more likely to be diagnosed with gastric cancer, but also they tend to be diagnosed at an earlier age and with more advanced tumors, in part because they have high prevalence of these risk factors, Epstein-Barr virus and Helicobacter pylori. So in the US, gastric cancer is not a common disease, but it's a common cause of disparities. And this is data in Latinos. These are the top incidence and mortality disparities in Latinos. So on the left, I'm showing you data for incidence. In the top disparities related cancer in Latinos are liver cancer and stomach cancer. So Latino men compared to white men are 71% more likely to be diagnosed with liver cancer and 47% more likely to be diagnosed with stomach cancer. In terms of mortality, gastric cancer is the highest mortality disparity. If a Latino man is diagnosed with stomach cancer, he is 2 times more likely to die than a white man. Same for women. In women is also the top incidence disparity. Latinas are 2.3-fold more likely to be diagnosed with gastric cancer, and when they are diagnosed with stomach cancer, they are 2.5 to 150% more likely to die than white women. So this is just the most important message here is that in the US, the most extreme cancer disparity in Latinos is stomach, in terms of incidence and in terms of mortality. We have also done some work on cancer epidemiology of Latinos in the US in terms of gastric cancer, and we published this paper in Cancer Causes and Control a few years ago, where we analyzed data from nearly 4,000 Latinos and 4,500 whites that were diagnosed with gastric cancer and reported to the population registry of the state. And even though people think gastric cancer is the same disease, the epidemiology of gastric cancer in Latinos seems to be different to the epidemiology of gastric cancer in whites. So let's just, okay. So for instance, more female patients are diagnosed among Latinos than among whites. Around a quarter of the cases are diagnosed in people who are 50 or 50 years or younger. A good fraction of the cases are diagnosed in people of low socioeconomic status. A high fraction of the patients have this diffuse histology, which is more aggressive, and a higher fraction of the Latino patients are also diagnosed with metastatic disease. So in general, the etiology seems to be different, and the clinical and pathological manifestations indicate or support what we see, which is they have characteristics that are associated with a worse prognosis or worse outcomes, particularly having more metastatic disease and more diffuse tumors. These are very difficult to treat. Okay. However, no, gastric cancer is not only an issue in Latinos. It's also a big issue in Asian American and Pacific Islander Americans. This is data from an analysis published by a Stanford group where they highlighted the tremendous heterogeneity that happens among Asian Americans. So Asian Americans are a highly diverse community, Pacific Islanders, in terms of immigration status, foreign born and US born, but also the diversity of the countries that these communities come from is incredible. And here in the US, for instance, the Koreans have a particularly high incidence of gastric cancer. So there are a lot of kind of differences in incidence and mortality in these communities that, I like this paper because it addresses also the need for people to work on disaggregating the information in Asian Americans. So, more research focus, research efforts are more focused on particular problems in these communities. So it's an issue in Asian Americans too. In whites, it's also an emerging issue. This is a paper from the NCI group, from Constanza Camargo, led some of this analysis where younger women are having an increase in incidence of particular subtypes. I think this is the body, cancers in the body of the stomach. And one of the possible etiological elements involved, or does not fully confirm, is the increase in autoimmune gastritis, which is a gastritis type that is different to the one caused by Helicobacter pylori. So then again, Latinos are affected, Asian American affected, also whites are affected. And as I mentioned before, depending on the gastric cancer type, on the location, for instance, non-cardiac cancers, the incidence is lower in whites and higher in minorities. But when it comes to cardiac cancer and esophageal cancer, which has a slightly shared etiology with upper, the cardiac is just next to the esophagus, the incidence and mortality is higher in whites. So if you think about gastric cancer, it's heavily affected by race and ethnicity when you stratify by location, right? And there are, I would say, disparities in every racial or ethnic group in the country. Another element that is affecting the epidemiology of gastric cancer is the obesity epidemic. The incidence of obesity-associated cancers is rising dramatically in the country. This is a study that was published a few years ago in the Lancet Public Health. And this is an example of gastric cardiac cancer. So this is basically the upper part of the stomach where the incidence has been increasing in younger people, but decreasing in older people. So it's being noted in most of the obesity-related cancers where the incidence is increasing in younger people. So basically this vertical line is, 50 or younger, 50 or older. And if it is above the horizontal line, the incidence is increasing. So as you see, for stomach cancer, incidence has been increasing, possibly associated with obesity. Okay, so that's kind of a summary of the epidemiology in few slides. We are doing some work on this. We are developing high-risk cohorts in the US and Latin America. So with funding from the NCI Center for Global Health, we have a supplement where we are testing two cohort approaches. One of them is we are developing a cohort of fertile relatives of gastric cancer patients. What is the rationale for that? One of the if you have a patient with gastric cancer, your risk of gastric cancer is around twofold higher than in the general population. That's the familial cohort. And another cohort that we are developing is people with pre-malignant conditions with atrophic gastritis and intestinal metaplasia. Those people have also an increased risk, and we are trying to develop these cohorts and see if we can follow them longitudinally to assess some of these, risk factor and identify possible biomarkers associated with progression from premalignancy to cancer. So that's another study we are doing. We have a cohort here in the US where we are focusing on epidemiology and genomic changes, somatic genomic changes in people with intestinal metaplasia. In those, we are analyzing not only the biopsies from the stomach, but also we are paying attention to the microbiome because we are seeing an incidence, possible role of bacteria in increasing mutagenesis here. And in collaborations with colleagues in Latin America, we are also doing some analysis of the Helicobacter pylori genome. The Helicobacter pylori strains are very diverse, and it seems quite population specific. So we want to see if the interaction of the germline genome in Latinos and the Helicobacter pylori, there is an interaction that increases risk. So that's kind of what is going on epidemiology-wise for gastric cancer in our group. Okay, so now let's move on to talk about genetics. So probably Climbot is more interested in genetics. And the classic familial form of gastric cancer is called hereditary diffuse gastric cancer. It's caused by mutations in this gene that was reported around 25 years ago, called CDH1 or E-cadherin, right? And these are family. This is a autosomal dominant disease, that is associated with diffuse gastric cancer. You know, there are two forms: intestinal diffuse gastric cancer, which is very aggressive, and also with lobular breast cancer. So this is a syndrome that every cancer genetics clinics offer it to people presenting with family history, fulfilling the criteria for this. So it took 20 years, and there were no new genes, at least moderate to highly penetrant genes. We wrote a review a while ago on this, and there are the classic CDH1. There is another one, which is CTNA1, which also seems to be associated with the syndrome. And gastric cancer happens as part of other syndrome. So people with FAP, with mutation APC, some of them have high risk of gastric cancer, as well as people with who have hereditary breast ovarian cancer syndrome or other kind of cancer syndrome. So gastric cancer is kind of part of the spectrum, but it wasn't clear what the risk was for them. Still remain not clear, but we are making some progress on that. There have been also efforts to identify common low penetrance variants for gastric cancer. Most of this effort has been done in Asia, in China, Japan, and Korea. And there are few SNPs with relatively decent odds ratios. And something that we are doing here too is to see if we can put together these variants into polygenic risk scores, and see if they can aid in stratifying risk among individuals. Around 5 years ago, we carried out and reported the results of an whole exome sequencing study in people who had family history but were negative for CDH1 mutations, and we found a high prevalence of mutations in these three genes: PALB2, BRCA1, and RAD51C, which are very important cancer genes. They were kind of suggested to be associated with the disease in the past, but this one was a study that focused on gastric cancer patients, and we found this. We reported this. Before we reported, we had a lot of back and forth with the journal, because it is possible that you may have gastric. One issue was that it could be phenocopies, that they were part of these families, but they have gastric cancer because other issues. For instance, they have Helicobacter pylori. We test, at least for the sample that we tested, they were negative. And also something that we did is we look at the tumors. We sequenced 3 or 4 of them of carriers, and they had the most important mutation gene that was BRCA, a homologous recombination deficiency. So that kind of indicated that there was a kind of causal relationship between the gene mutations in these homologous recombination genes and the fact that they were having gastric cancer. Then, fortunately, more and more studies are reporting association between these genes and gastric cancer. So PALB2 is one that has been many more studies are reported patients or families with PALB2 and gastric cancer, as well as ATM. So this is slightly outdated, but ATM is also coming up quite frequently, as well as some of the other BRC genes. So one of the, I think one of the most important advances on gastric cancer genetics in the past few years has been probably the fact that homologous recombination is emerging as a pathway that may be harboring gastric cancer predisposing genes. So with this paper, we actually wrote another one, and we were lucky to have funding to get this other one, where we we have many more families. So this is not a fully analyzed data set, but we are at the moment analyzing data from 500 families or patients with early onset disease. So in this study, we are sequencing anyone with gastric cancer with first or second degree relative with cancer, or gastric cancer patients that were diagnosed before age 50. And we are, when you do this exome sequencing study, you find ton of mutations. So we are kind of prioritizing this. So we are looking initially at the, known high-risk cancer genes like ATM, ATR, BRCAs, PALB2, etc. And then we are kind of going down in the analysis. We are just trying to complete this analysis. This is data from the type of variant we found. I talked with Sharon about this in the past. We are doing a lot of BUSs, a lot of variants that we don't know what they are. So I think interacting with ClinVar will be great in this aspect. An issue is that there are a lot of new genes where there's not much information. So I mean, the annotations for the well-established genes are good, but for some of the other ones, we need to do more work. So we have, if we look at ClinVar, we have 475 pathogenic variants in 39 genes. I don't have the full data here, but the prevalence on highly penetrant cancer genes like BRCA1, etc., is around 7%. Okay. So in the past, and when I show you, people used to kind of, I wouldn't say dismiss, but people used to think that gastric cancer was a mainly environmentally caused cancer. This data and data from other studies are supporting more and more that a significant fraction of the patients are indeed the result of having an inherited predisposition to the disease. So there are some new genes here that we are replicating in data from Genomics, TCGA Biobank. So this is, for instance, BRCA1. We are seeing some association at the gene level. There are some other genes that I'm not at liberty to report back because this is data from us and collaborators, but we are hoping to have a paper reporting this soon. So we are seeing some indication that we will have new genes. So we are working in two areas now. One is to do a lot of validation of these findings. So we are doing co-segregation analysis, we are doing somatic analysis, etc., to gather evidence for the most important genes. But the first paper that you are going to see from our group is just reporting the prevalence on the known genes. So we are actually, this is an international consortium. So we have studies in Europe, Latin America, and us. And we would like to report the prevalence of the mutations in genes like BRCA1, BRCA2, ATM, PALB2, as well as the characteristics of those families. So hopefully that would be useful for the field. I think, it's important to know the prevalence, but also it's important to have some initial estimation risk, cumulative risk, and that's what we are going to do in the publications, which I'm hoping it will be out soon. But, because we have all this bunch of genes that, for which the evidence is not, we need to provide more evidence. So something that we are doing in the lab is, I lead another study, which I'll mention later, which is to make patient-derived models for minorities. So we are making patient-derived xenografts and patient-derived organoids from a study in California and in Texas that is morely focused in minority patients. So we have some normal organoid lines from Latino patients that we are modifying using CRISPR-Cas9. So I think this is an example of, let me see, what is this? I think it's PALB2. Okay. Oh, this is result here. So we actually have a line where we have a dual mutant of T53 and APC. Okay. That is, we are doing a bunch of analysis to see if we are hitting APC. So are we increasing Wnt signaling? We are increasing Wnt signaling. We are increasing a lot of genes that are, in the pathway. So the effects of the genetic manipulation in these normal organoids is what we expect to see. So in this case, TP53 and APC, we see an increase in Wnt signaling. But interestingly, we are also seeing that the organoids are kind of having phenotypic modifications consistent with kind of pre-malignant conditions, right? And also the staining of beta-catenin is located to the nuclei, right? So which is what we see in patients with general mutations or somatic mutations in Wnt signaling genes like APC. So we knock down APC, beta-catenin goes directly to the nuclei, and we are seeing some morphologic changes. So we are kind of reconstituting cancer in vitro using models that are derived from minority bases. So that's kind of part of some of the work that is ongoing to validate some of these hits. I showed you some example of one on gene here. Okay, so now let's move on to somatic genetics. As I mentioned, there are some classifications of gastric cancer. One is based on location, cardia and non-cardia. They have different epidemiology. Another classification is based on histology, the Lorentz classification, where some tumors are classified as intestinal, some as mixed, and some as diffuse. And then, nearly 10 years ago, the TCGA came with a new classification, which is a molecular classification. And they based on multi-omics studies, they found 4 subtypes: Epstein-Barr virus, the microsatellite stable subtype, genomically stable subtype, and chromosomal stable subtype. And there were some associations with clinical characteristics. Some of them were more common in men, some of them were more common in women, some of them were common in young patients or older patients, and also some of them were common in certain parts of the stomach. And as well as some of them were target rich, i.e. they have a lot of mutations that could be targeted with therapies like, PIK3CA in Epstein-Barr virus tumors, or RTK RAS amplifications in chromosomal unstable tumors. So the TCGA was good to come up with, another kind of way to classify tumors, and there was some clinical and AP associations that were interesting there. However, you probably have seen this like multiple times, but the TCGA has a major problem, is that TCGA have very few minorities. Okay, so this table doesn't even include stomach cancer, but you can see the number of tumors from Hispanic was really low, right? So gastric cancer is the top cancer health disparity in Latinos. The TCGA just didn't include Latinos. Okay, so that's a major problem. So if we want to know more about the molecular epidemiology of gastric cancer in Latinos, we don't know it. Also, we don't know if they are target rich or target poor, or if they have particular mutations that could be used for developing new therapies, etc. So that was something that we are working quite hard in the lab. The other one that I mentioned, the general genetics, is also another aim, is to sequence around 200 tumors from Latinos and do like a gastric TCGA in Latinos. That's what we are doing this year as part of the other one. However, we have done some work before. So we published a study in Cancer Communications a few months ago, where we look at intratumor heterogeneity. And intratumor heterogeneity, for the study, we took we use a method called multi-regional sequencing, where we took biopsies from tumor we took multiple biopsies from the same tumor that were kind of separated by at least 2 centimeters, and we were wanting to see which mutations were common between all of the biopsies. We call those clonal, and which ones were not common. We call them private or subclonal. But also, because we have this data, we estimated the prevalence of the subtypes, the molecular subtypes. So these are the 4 molecular subtypes: chromosomal unstable, Epstein-Barr virus positive, microsatellite stable, and genomic stable. I forgot to mention genomic stable is the most aggressive type, more common in diffuse tumors, and is chemo resistant, doesn't respond to chemotherapies, has very few targets. So there are not many targets that we can kind of use drugs to treat them. And also they are immunologically cold. So these are the worst cancers to have. Well, actually, we found that in Latinos, nearly half of the tumors from the Latino patients that we analyzed had the most aggressive subtype. Okay, a study published by one of our colleagues in Texas, in Dallas, found the same in Texas and Latinos. So these are mostly Mexican Americans in California, Latin Americans. So the message here is that we believe that a possible molecular cause of disparities part is that the Latinos that we are seeing, or the Latinos when they are diagnosed with gastric cancer, a lot of them have the most aggressive subtype. So that is an area where we are quite interested in researching here in the lab. Actually, we're doing a lot on this aspect. We also did this analysis of how mutations, these are the, a lot of the genes that you call, druggable, so say RBB2, HER2, in a good fraction of the patients, those druggable mutations are not present in all tumor cells. So we weren't wanting to investigate, what was the patterns of that. So let's start with genes like TP53. Is that tumor suppressor genes? There are no drugs or approved drugs to treat TP53. Normally, when these are, let me explain this. This is a complex table, but these big columns are patients, and every when they are we actually sequence, every column here is a biopsy of a patient, okay? And the darker lines separate one patient from the rest. For instance, for the first patient, this patient has 2 biopsies, has an Epstein-Barr virus positive Epstein-Barr virus subtype, and it has a clonal mutations in RBB2. We did this for multiple cancers in multiple biopsies, and we found a lot of heterogeneity on these druggable genes. Okay, so we believe that when you treat someone with a targeted therapy, ideally you will expect that patient will have the target in every tumor cell. Okay, what we are seeing here in this data set is that not all targets have clonal mutations. So you may hit some of the cells with the target, but some of the other cells that don't have the target may come back and grow. So this is quite an important element to consider in targeted therapies. So when you send the patients for, like, say, foundation medicines, say, oh, they have the target, yes or no. It's not only important to have the target, it's also important to know if that target is clonal. Okay, so the expectation is that may work, the therapy may work better than if it's clonal or not. Okay, and also something else that we look at was the prevalence of druggable mutations by subtype. Microsatellite stable tumors, right? They have a ton of mutations because they have a hypermutant phenotype. They have more targets than genomic stable tumors, where only 1 in 5 have had a mutation that we could say it can be targeted. So the message here in this part of the presentation is that Latinos have a different molecular subtype profile. They are enriched by the most aggressive subtype, and that most aggressive type also lacks targets. So it's actually quite difficult to develop precision medicine in these genomically stable tumors. Something else that we look at is, because we know which mutations were clonal and not clonal, we look at mutation signatures, right?
Luis,
So
I want to make sure we've got some time for discussion. So.
Okay, this is I think the last slide. Yeah,
Okay,
this is the last slide.
Okay,
Okay, so basically we look at the mutation signatures, and we saw things that were new, like aflatoxins. We saw an effect of aflatoxins in gastric cancer. We don't know if it's real or not, but there could mean a new risk factor for gastric cancer. And for instance, in tobacco, in the microsatellite stable tumors, we saw most of the tobacco mutations were clonal. They actually, if you smoke, this mutation will initiate cancer. In the MSI tumors, they accelerate cancer. So they're more common in the subclonal status. Of course, this was data from 30 patients. We would like to replicate this in a larger study. Okay, so last slide. Gastric cancer is the leading cause of morbidity, mortality, and it affects all populations in the country. It has a particular high burden in minorities. A significant fraction of the patients has an inheriting etiology, which is important to consider for prevention using genetics. And this extensive heterogeneity is important to consider because it affects response to therapy. These are my acknowledgement, my lab, my collaborators, and the funding. These are the people who actually do the work, so they are the ones who, some of them should be presenting here. I'm just do this. So thank you for the opportunity. I'm happy to answer questions.
Louise, great. Thanks so much. So there was one early question and then I have a question, but please others put them in the chat. So Aaron Ramos, who's our program officer, pointed out that one of the things we're really trying to do is make sure that variants identified in underrepresented populations are being put into ClinVar, and those can be picked up. Like, as you know, we have a CDH1 expert panel that could evaluate them. So are the variants coming out of your work? Because research variants can also, classifications can also be submitted. Are they being submitted to ClinVar?
This is like, we are analyzing data. At some point, they will. We also are going to deposit this in the VCAP. But yes, I think that would be, it would be good to work with you. Something interesting, actually, I'd be like, we didn't find anything much in CDH1. CDH1 is very rare.
Yeah.
Okay.
Right.
So we don't, I mean, I can tell you, I don't think we have found a pathogenic like pathogenic mutation in CDH1. I will need to double check, but I was surprised to see that. Okay. But we will look at the kind of the other categories.
Right, but the other genes also have expert panels, so
yeah,
it'd be great.
Yes, and actually we are stratifying the analysis. So we have around 300 of the patients are from South America and 200 are Europeans. Guess what, we are seeing more BUSs. So we are actually developing a study on that too. Yes, but at some point we would be submitting them to ClinVar, and the data is, will become available in DBGAP soon too.
So I have more of a philosophical question. So it was really disappointing, and not everybody on the call is familiar with TCGA, but it was by far NIH's largest investment in tumor sequencing, at the time, starting in around 2014 or 15. So it was very disappointing to see your slide about how few Latinos were included, even though gastric cancer, has such a large component in the Latino population. So do you think that so this is the philosophical part, do you think that there is enough awareness in play now that if someone were to develop a TCGA today, that wouldn't have happened? Or do we still really need to be very specific when we're developing cohorts to make sure we're including the relevant populations?
I think that the time of ivory tower genomics is gone. I think, this wouldn't happen now. There was happening I mean, we have been working in disparities for a long time, but disparities became, I mean, like, one of the silver linings of the pandemic is that people talk about disparities more and more than before. People didn't realize there were disparities. I mean, unless you were working in a specialized field, we were kind of a fringe group working on disparities, right? But it is, I think everyone is fully aware of the need to advance disparities and to advance opportunities for minority scientists. So I think, if you send a grant to the NIH studies and you don't have diversity, you get dinged. Those studies are not. I think those studies are part of the past. A big requirement for us is to make, these studies inclusive, right? And because they have implications for many, precision medicine, prevention, etc. So it is, we have been actually fighting a lot for this. We have been quite vocal in the past few years, and I think now NIH and all the kind of private organizations are diversity is at the center of many of these organizations. So, yeah, it's a pity that it didn't happen 10 years ago, right? But, well, we are kind of doing that, and there are many more groups that are kind of filling the gaps. There are efforts on multiple cancer types, the efforts on breast cancer in Black women, triple negatives, a lot of effort on that. Prostate cancer in Blacks, we are leading some of the efforts in gastric cancer in Latinos. So I think, the situation is changing, and but yeah, it's unfortunate that it didn't change fast enough. I see some.
So there was a question about engaging Latino colleagues in our ClinGen curation activities, including those based in Latin America, as well as, of course, in the US.
Yeah, I encourage you to do it. People bring different perspective. It's I think the groups will be enriched if you diversify your team. So think about, and if you are senior, think about supporting and mentoring junior faculty who are diverse. So, I will encourage clean bar, and everywhere I go, I just encourage to bring more people that bring different perspectives. And there are a lot of great people out there, I tell you.
Yes.
So the excuse of, oh, the pipeline is good enough. There are I have met a lot of great Latino and Black scientists. So think about them.
Yeah, so maybe I'll follow up with you by email and maybe get the names of some organizations as well that we could ping. And then there was a more specific question about, obviously if you're finding BRCA1 or BRCA pathway variant in the gastric cancer families, do they have the typical BRCA tumors as well?
I cannot tell you for certain, but some of them do, some of them don't. Okay. So I think we are seeing this in Latin America because there is also a high incidence of gastric cancer. There may be an interaction between, of Helicobacter pylori.
Yeah.
So there may be an interaction. So I think we don't see this in Europe because the incidence is low, right? But this could be a true gene environment interaction that is modifying the risk of these patients. But it wasn't obvious. I mean, we enrolled them. That question from we enrolled them because they had gastric cancer, right? So that's something that we're very interested in investigating. Why do they have gastric cancer, right? Because not all the BRCA families have gastric cancer rates. I think that could be important for all those families, right? Because maybe some of them are recent, they don't know way to tell, right? So we're thinking in developing studies that address that particular aspect. The risk of gastric cancer in BRCA families ascertained with ovarian and breast cancer patients, as well as the risk of gastric cancer in these families. And we are going to see, if we work out or come up with biomarkers or ways to predict risk. Because this is a bad one to have. Gastric cancer has a bad prognosis.
Yeah. Well, and it was striking your more recent work that not only is the risk higher, but the molecular subtype is worse.
Yeah.
I think it's an interesting question whether it's an environmental, again, most of the classic breast cancer, BCAC, and the other large cohorts are almost entirely European. And so whether it's an interaction, European and US and Australia, so whether it's actually an interaction with like H. pylori or something would be really interesting.
Yeah. We are trying to design a study that, but for those, we need to design the study because we kind of started with the patients, right? So, and this is also, we need to have more diverse cohorts. It's difficult to do some of these studies. We need to actually develop the studies ourselves. It's not like UK Biobank. All of us is having some diversity, I have to say. But yeah, I think an interesting question in the field is, we're showing that these genes are likely to be cause of gastric cancer. How can we tell who has risk or not in thousands of families with BRCA mutations? That's something that we would like to get in. We are actually trying to answer these questions.
Any other questions for anyone? We're almost at the top of the hour. You mentioned the druggability of the tumors, but have you, since I know Terry's on, has there been any work related to pharmacogenomics itself? Do the patients have any variants that make them less likely to respond more commonly in the Latino population?
We have another study that I didn't present in detail here, which is I have a new 54 where we are developing precision medicine in minorities using ethnic or race appropriate models. Those questions, we could answer them as part of that. At least for targeted therapies, it's important to, if there is something that is in the genome of these patients or the ancestry of these patients, we should be able to see it. Because also, if you look at the models, most of the cancer cell lines are from whites and to certain extent from Asians. So there are not many cell lines from Blacks or Latinos. So in that study, we are trying to bring that component to the precision medicine experiments.
Yeah, I think that's something we don't think nearly enough about, which is what is the underlying. I noticed your work on the organoids. Yeah.
Yeah. So we have that grant. We are trying to renew it. So next month, hopefully we'll have good news on that one. But yeah, this actually it's quite an interesting collaboration, and we are we have a lot of more models on gastric cancers that are being sent to the NCI. I think we are sharing them. So, you know, if there is a disparity affecting a particular group, we need data from them, right, to find what are the characteristics, and also we need models to address those questions in race, ethnic appropriate models.
Right. Okay, well, thank you so much. And thanks for participating. And I'll get back to you about some of the other things about the workforce and getting some ideas from you. So
Yeah, happy to. Thank you very much. Have a good day all.